Let me first welcome again Dr. Andrey Klymchenko. Sorry if I don't get your name correct.
It's all right.
Yeah.
It's all right.
So he obtained his PhD from Kyiv National University in 2003. He made postdoctoral studies in the University of Strasbourg from 2003 to 2005 on fluorescent probes, and in Catholic University of Leuven on supramolecular chemistry in 2005. So he joined CNRS in 2006, received CNRS Bronze Medal in 2010, and became a group leader in 2015. So he obtained ERC Consolidator grant BrightSens in 2015 to work on fluorescent nanoparticles for biosensing. He's a co-author of more than 220 peer-reviewed articles and the co-inventor of 10 patents. Very impressive record. All right. So let me give you the abstract for today's talk. So the title for today's talk is Bright Fluorescent Probes for Biosensing and Bioimaging, from dyes to nanoparticles. So fluorescent techniques rely on the performance of chemical tools, which most of us here refer to as probes. Tailor-made fluorescent molecular probes can change their color via, these are called solvatochromic dyes, or intensity fluorogenic dyes, in response to cellular targets. We are talking about biomembranes and organelles, and also biomolecules, receptors, and RNA. Brightness of probes can be boosted by assembling the dye-loaded organic nanoparticles. Concepts of polymer nanoprecipitation and dye insulation with bulky counterions enabled preparation of 7 to 100 nanometers nanoparticles. These are more than 100-fold brighter than semiconductor quantum dots, and application for bioimaging and amplified detection of nucleic acids. So let us welcome Dr. Andrey Klymchenko to give us the talk today. Thank you.
Yeah. Thank you very much, Ken. Thank you for this kind invitation and nice introduction. So I'm happy to be here. So hello, everyone. Yeah, so today, as Mr. Chairman presented, I will be talking about new fluorescent tools, and I will show you the path from dyes to nanoparticles. And my team works on three big research directions. First one is molecular probes. Here we work on different molecular designs to achieving targeting biomembranes and organelles, detection of RNA and receptors, and this is all in live cell imaging. The second direction is, I'll probably move a little bit our photos. Yeah, so the second direction are fluorescent nanoparticles. Here we work on different designs I will be talking about, and the main applications are biosensing, especially for diagnostics application and detection of nucleic acids. And also we work on the bioimaging using nanoparticles. I will show you some examples. And some emerging direction are fluorescent biomaterials, in particular near-infrared materials for image-guided surgery, and so in different forms, in form of coatings or nanofibers. But this I will not talk about. So I will talk about these two big directions. So why do we, so I just want to move a little bit the screen and the presentation screen to see the titles up. So why do we work on fluorescence? This is a unique technique because on one hand, it is sensitive. You can go up to single molecule detection level. You go to picomolar, even femtomolar concentrations. It is very fast. Because the fluorescence lifetime is very short, you can go actually down to nanoseconds, even femtoseconds speed of detection. Then it is non-invasive because you don't touch the object, but you rather shine light and record something, and it is specific because you go down to molecular resolution. Because to see something, you can address individual molecules, but for that you need fluorescent probes, fluorescent tools. And then you can go from single molecule to cell, tissue, and up to the whole animal imaging. And So the concept of fluorescent probe that I mentioned, it's a molecular nanoparticle that goes to the live object. It can be cell or tissue, and then you need to excite it with light, and then it provides you fluorescence. And from fluorescence, from its intensity, spectrum, ratio of intensity, lifetime, or isotropy, you can get an information about your biological object. You have two types, principle of fluorescent probes. On one hand, you have fluorogenic probes that provides you turn-on response to the target. So the molecule can be non-fluorescent in solution, and once it binds to the target, RNA, protein, or membrane, it turns on. Or it can be a chromogenic system which changes the color. So it can be blue in solution, and when it binds to the target, it becomes red. And the probe usually is composed at least of two parts. You have a reporter and you have a targeting group, but then you have probably also the
I'm sorry, Andrey.
Yeah.
I think your slides is frozen. Are you on? We are looking at your fluorescence slide.
Okay, just a moment. Something happened.
So sorry.
Okay. I don't know what happened. Just up. Now, is it okay?
Yes.
I don't know what happened. At some point, probably I was changing something and it got frozen. So, do you see the, yeah, okay. Laser pointer. So the fluorogenic response I was mentioning. So here you see the concept of fluorescent probe, smart molecule. Yeah. Hello. Yeah. Okay. So fluorogenic response is a turn-on, so it's light up system, and then chromogenic response is when the molecule changes the color when it binds to the target. And so the fluorescent probe is usually composed of reporter, a fluorescent molecule, and a targeting group, but then you can have also a transduction system, the communication between targeting group and the reporter. So now the principle of probes, you can have many mechanisms of detection, and the one that we exploit a lot is based on environment sensitive dyes. And here you can have different mechanisms that in principle what happened that in solution, usually it is water, especially in biological imaging experiments. So the molecule can have be non-fluorescent or one color. When it binds to the target, it changes the color. Why? Because in the target environment, it can be apolar or viscous, and this changes in environment can drive changes in the fluorescent properties of the dye. So there are many different mechanisms that drive the response of probes to their environment, polarity or viscosity. One of the most common are excited state charge transfer. This is based on the push-pull dyes, where the color changes as a function of polarity. You can have excited state intermolecular proton transfer, where in the excited state proton moves from one side to another. These are three hydroxy from one family. And then the dual emission of these dyes depends on polarity. You can have twisted intramolecular charge transfer, where the molecular rotation changes, quenches the fluorescence of the dyes, and then intensity depends on viscosity. You can have also the ground state rotation, and these are so-called flippers, recently drawn this, where the rotation or planarization in the ground state may lead to strong changes in the excitation, in emission lifetime, etc. And then another remarkable example are aggregation post-quenching behavior, where the response is driven by aggregation or disaggregation of molecules. I will present you actually three examples of these mechanisms. So let's start with the chromogenic probes based on solvatochromism, so those that are sensitive to polarity. In principle, how they work, these are push-pull fluorophores. On the excited state, they become highly charged, so highly dipolar. And therefore, they strongly interact with the environment. So that, for example, in water, the strong interaction leads to the decrease in the excitation energy compared to the oil phase. And therefore, when you increase polarity, the color changes from blue to red. So the spectrum shifts to the red. And if you look at the fluorophores, which are sensitive to polarity, these are all push-pull molecules. So you have a electron, you have the electron donor group, and then you have an electron acceptor group, which is in blue. And actually, we worked before a lot on different fluorophores, and in particular, for example, small molecule like fluorine, by adding push-pull groups, it can become strongly solvatochromic, and compared to other solvatochromic dyes, which was one of the most sensitive. And here you have an example for picture in UV light, where the color changes from toluene in blue to methanol in which you appear in orange. So, and in fact, in this scheme, at the moment we worked on this project, there were only blue and green dyes, absorbing actually the wavelengths around 400, 450 nanometers. And we were interested to shift to the red. In fact, non-red was the only exception of a red polarity sensitive dye. And we were interested in near-infrared dye. And here's an example of a molecule, it's called deoxybarine. Which remarkable property is that its acceptor is complex with boron, and in fact it drastically increases the emission properties, quantum yield, and it has a long push-pull structure. And this kind of molecule actually absorbs already in red and emits in near-infrared. It has a very strong variation of color, which goes up to 800. It was actually one of the first examples of a dye emitting near-infrared and presenting this property to response to polarity because of this push-pull system. But why I'm talking about this polarity sensitive probe? Because they are actually perfect tools for detection and imaging. And the first example of biological application are membranes. So membranes, as you know, is a lipid bilayers. And inside lipid bilayer, the environment is highly apolar. And therefore, what happened when a molecule like non-red binds to the bilayer, it changes the color, as I was explaining before, from highly polar environment where it is red to a highly apolar environment where it is blue. And this is what you see in the emission spectrum, but also it lights up because the quantum yield in a polar environment is much higher. And this change in the color is actually a fundamental property of these molecules that we can use for sensing. And how? The example is to detect organization, self-organization of lipids on the membrane. As you probably heard about, the membrane is composed of large amount of different lipids. And around 2000, there was a hypothesis of so-called lipid rafts that actually certain lipids, especially unsaturated lipids and cholesterol, are organized together in tightly packed structures, which they call rafts, which swim in their liquid phase of unsaturated lipids. And the difference here is this is tightly packed structure where the molecules are just next to each other, tightly packed, and they don't move much, and very loose structure. And so the hypothesis is that when you put a solvatochromic molecule in the system, here in tightly packed structure, you have no water because there is no place for water. And in the loose structure, you have large quantity of water. So the color of the dye should be different. And but to do this, so what we need to place environment sensitive dye specifically at the membrane of the cell. And for doing so, we took a non-red. So we will see a lot of examples of non-red derivatives that we synthesized. And what we did, when you actually put non-red to the cell, it doesn't bind to cell membrane. It binds to lipid droplets and intracellular structure. And these are two cells, and you see these dots, these are droplets, and all the rest is intracellular environment, usually membranes, and they're in different color because polarity of droplets is lower than other components. So to target membranes, especially plasma membranes, we need an anchor group. And in particularly, we took a lipid-like structure. You have a zwitterion group and long chain. And when you add it to the cell, you can see a nicely delimited two cells. The labeling is very specific. So these are early studies, just to show you how does it work. And in fact, when you go from ordered phase, which is saturated lipids and cholesterol, and disordered phase, you see a strong change in the emission color. There's a shift. So low hydration, liquid over the structures is shifted to the blue. So now we want to image that. We want to see it in color, this membrane. What we do in this case? In the microscope, we have to split emission of this dye into two channels. The blue channel, which is this part of the spectrum, and the red channel, which will be this part of the spectrum. And then when there is a shift, the ratio between this part and this part will change. And this is what we do when we image model lipid membranes. These are so-called giant vesicles. They can be prepared by electroformation. And these are models, synthetic models of biological membranes. They can be the size of a cell. You can tens of to 50 microns. You can make them. And so when you look at their at this structure, you can see that the color changes as a function of ordered and disordered phase. So in ordered phase, the ratio changes so that because of this shift to the blue. Now, if you, so this actually the color is a code for the ratio of these two bands. Now, if you take mixed phases, in the mixed phases, you can see both phases present and you can decode them by the color, and color which represents the ratio and ratio represents the shift. So now we can actually see those rafts, but on the model membranes. The problem is that when you go to the cell, you don't see that. In the cell, the cells are, so you have several cells here. So cell is like this, and the membrane is very complex, but the color is more or less the same. You don't see these rafts, you don't see these lipid domains. And in fact, the problem when we did this, we look more carefully into literature and we realized that those structures are very small. And fluorescence microscopy has a limitation, which is about 300 nanometer lateral resolution, so that you cannot really reach visualize these small structures. And for that purpose, you have there are new methods which appear, which are called super resolution imaging techniques, where, for example, based on single molecule localization microscopy, taking a dye that goes from off state to on state, you can extract super resolution information. How they do it? You stain the cell like this, and then you shine a strong light, and then molecules start to blink. They turn off, and then on the image you see only a few points. You localize these points, and then you record at least 10,000 frames, and then you reconstruct the image. In fact, it looks like this with a little bit of French taste. You start recording the blinking events, then you localize each spot with a nanometric precision, and you reconstruct the image, and you get this beautiful image of it. Okay, here it's not nanoscopic, it's a little bit big, but it worked well on the cells. So in our design, as you remember, we had a probe which binds well to the membrane. However, when we do this approach with this probe, with high affinity to the membrane, we cannot have any blinking process. It's very few blinking events. So this was done in collaboration with Kyiv in Berkeley, who helped us to do this single molecule imaging. And therefore, what we did, we redesigned the dye and we changed the length of their hydrophobic chain. We also changed the charge. We actually inverted also the modification because we found out that there was a problem of photostability. So we removed this phenolic oxygen and we make substitution from amino group. So we redesigned a lot of the dye. And then we had two dyes, long chain and short chain, hydrophobic chain. The hypothesis is that here we will have strong affinity, here we will have low affinity. Low affinity to membrane means that the dye will go in and out, in and out. And when it goes in, it binds, it turns on. And when it goes out, it turns off. And therefore, when you look at the image, these dots are single molecules, and these are events of binding and unbinding because the process is reversible. And in case of a short chain, you have a very, very fast processes of binding. In case of long chain, the process are much more rare because actually it binds irreversibly and bleaches. It works also in case of NARED, but NARED is not good because it goes inside the cell, so we cannot really image well the membrane. So now when we go to the cellular imaging, when you put a high affinity probe that binds irreversibly, you see nothing in super resolution imaging technique. However, when you have a reversible probe, then you can get a beautiful super resolution images of the cells, and you can actually start imaging some details of the structure of the membrane then you can follow processes like endocytosis with nanometric precision. But you can do more than that. In fact, using a prism, from each dot, you can extract color information. So each dot is actually a spectrum, which you can decompose using a prism, and you can obtain those little spectra for each cell. And therefore, you can reconstruct rate of the image where each pixel is actually a spectrum. So this was again done in collaboration with Kyiv in Berkeley. And so here he could make this spectrally resolved super resolution imaging SR paint. And what they showed is that each dot, or even any mechanical distortion of the membrane, leads to the redshift, which means membrane become more disorganized when it's bent. And this can be done with nanometric precision. Okay, this was about surface. Now what is happening inside the cell? Because inside the cell, you have lots of membrane structure. You have endosomes, endoplasmic reticulum, Golgi, mitochondria, lipid droplets, etc. Can we image them specifically? It can be obtained some information about lipid organization. And why also? The thing is that surface of the cell is contain nearly 60% of cholesterol. While, for example, in the plasmic reticulum, you have only 5% of total cholesterol. So there is a huge variation of lipid organization. And this variation is actually means that the organization of lipids inside the cell and outside the cell are totally different. And functional states like cancer, transformation, immune response will definitely alter all this process of stress, cell stress, etc. So what we did, we say, okay, we can target our Nile-red probe to plasma membrane, but we could, in principle, by using a targeting group, like, for example, morpholin group, which can be protonated, to localize it in lysosome because of low pH there. We could put also a hydrophobic group to be more specific to lipid droplets. Then charged group to go to mitochondria because it has negative membrane potential inside, it can attract. We can add also the reactive group, which is known to bind ER in the plasmic reticulum, and then the long chain to go to Golgi. So we synthesize all these molecules. And after that, so the synthetic synthesis is relatively straightforward. So in fact, the key step, once you create this acid, you need to get a nitroso derivative with orthohydroxy, and you actually condense it with a hydroxynaphthalene. And at this step, you create a Nile-red derivative, which is acid. And after that, we add different anchor groups. So, for example, in case of membrane, it was the secondary amine, or all other amines to add organelle targeting group. Okay, so as I said before, the molecule with anchor group binds to lipid membranes, and we verified it with a reference probe. So now for each organelle targeting group, so in this case, in the plasmic reticulum, we colocalize with AR tracker, and you can see good colocalization. We take mitochondrial probe, we compare with tracker, it localizes, colocalizes, then lysosome probe, you can see nice dots, colocalizing laser tracker. It's actually colocalized even better than commercial laser tracker. Then Golgi gives nice colocalization with Golgi tracker, and the same for lipid droplets. So in principle, by taking a neutral molecule, which is not too polar, not too apolar, and adding a ligand that targets given organelle, you can target specifically different intracellular structures. But you can do more than that, because our molecule changes the color as a function of lipid organization. So when we start imaging these structures in terms of the ratio of the blue to red, as you remember, I explained you, when you take a ratio of blue to red, which reflects the polarity shift, you can see that each of these structures represent different color. And this color is coding polarity. So more it is reddish, more it is polar. And so you can see that lipid droplets are the most apolar again, then membranes as well, and the most polar are ER structures, mitochondria, etc. So this was very important, and then we applied the cell stress. What could we actually, as one of the important stresses are oxidative stresses related to immune responses, inflammation, etc. So then we applied hydrogen peroxide, a very simple treatment. And with this treatment, we can already see that the color of lipid droplets, for example, lysosomes and mitochondria changes dramatically. What actually happened that there is a peroxidation of lipids, which increases local polarity, and the probes respond. But different organelles respond differently. Some are more resistant to oxidative stress. Membranes are more resistant, for example, than lysosomes or mitochondria. Okay, then we go to fluorogenic systems, fluorogenic system that turn on the light. And those, the first example I wanted to show are molecular rotors. So this is a different mechanism. It's no more push-pull, although this molecule is push-pull, but the key importance here that it rotates in the excited state. And in literature, how you can actually find many examples of those molecules. And a remarkable example is body P dye, where this phenyl rotation quenches totally fluorescence in normal solutions, non-viscous solutions. And then increased viscosity, the average fluorescence lifetime, but also intensity goes up. And then this was applied earlier to target different organelles and to visualize viscosity, local viscosity, for example, in mitochondria or in endoplasmic reticulum. Like we showed with polarity, it was shown for different organelles. But we wanted to do something else, and we actually wanted now to address a specific lipid. So before I showed you the lipid organization, so this means how they're tightly packed or not. But here we wanted to detect a very specific molecular species, and this was phosphatidylserine. And our hypothesis is that, okay, if we can have a targeting group that can recognize specifically a head group of phosphatidylserine, and we attach this molecular rotor molecule, turn-on probe, what could happen? Once it is recognized by the receptor, this viscosity-sensitive probe will be placed next to the lipids, because localization of their ligand will impose positioning of the fluorophore inside the lipid membranes, which is expected to increase viscosity. So therefore, we synthesize this big structure composed of the fluorophore and the receptor, zinc ligand, zinc-based complex. And in fact, we wanted, this guy is sensitive to viscosity, increase intensity with increasing viscosity, this is all right. And what we wanted to detect is apoptosis, because phosphatidylserine is exposed at the outer leaflet on apoptosis. So for that, we had to, it was a convergent synthesis. We needed to first to synthesize the body pi part. We used actually click chemistry to combine it later with our receptor part. So the receptor part, so it's a multi-step, but again, it's relatively straightforward synthesis, where you first synthesize this complexing agent based on this pyridyl structure, and then we put the acetylene group, and then we couple these two guys together by copper catalyzed click reaction to obtain just the starting material without zinc, and then we add zinc, we get the zinc complex. So this molecule then we apply to cells, and what we could see. So in fact, what we do, the cells are treated with a special drug which induces apoptosis. And then we also apply a probe that will recognize apoptotic cells. It's a reference probe, annexin. So everything which is red is apoptotic cells. And we could see that it colocalizes well with our zinc-based molecular rotor probe. So where it is red, it is also green. So this is an apoptotic cells. It is detected by our probe. But also what we noticed that our probe can also visualize actually late apoptosis, because it goes inside late apoptotic cells. So we could even distinguish early and late apoptosis. Okay, so this was example of a rotor. Now we go for another one which is based on aggregation structure, aggregation and disaggregation. So this is also fluorogenic systems, which are based on dimers of probes. And what we wanted to detect, we wanted to detect proteins now. And protein fluorogenic systems are actually well known. For example, these are called fluorescence activating proteins. They can activate structures like malachite green, which is non-emissive. And once it binds to a special pocket of a protein, it can light up. You can have also genetically encoded proteins which can recognize small molecules, like this fast tag structure, where it lights up, and so you can light up a given protein. But we wanted to do it a different way. We wanted to target a membrane receptor so that the fluorophore, the dimer, will be inserted in the lipid bilayer. And the hypothesis is that in water, the dimer is closed. And when it is closed, it self-quenches. Once it binds to the membrane, it opens up and it becomes fluorescent. And for that, we designed actually the dimer based on squarene. And these squarenes, they are relatively hydrophobic, and we put polar groups around to ensure solubility. And we had also the monomer. And so what happened when you put the dimer probe in water, you actually have a characteristic spectrum where there is this band which is blue shifted with respect to a molecular species when it is in organic solvent. This shifted band is the aggregate, so-called ash aggregate. It is not observed in the case of monomer. In case of monomer, you have a classical spectrum of the dye. And so when you change the solvent to a polar solvent, there is a shift because it opens up. In fluorescence, you can see when you add methanol to water, you have increase in the fluorescence. So you have a fluorogenic system that opens up when polarity changes from water to methanol. This is what we need because we'll go to membrane, which is apolar. And this is what you see. So what we had to, of course, to target membrane receptor, we took a ligand. This was cyclic peptide, carbitocin. It targets G protein couple oxytocin, G protein couple receptor oxytocin. And when we added to the cell, we could nicely see the membrane of the cell. However, when we took a probe, which is not a turn-on probe, it was a rhodamine-based probe for comparison, we added to the cells, we could see fluorescence everywhere, also on the membrane, but as it is not a turn-on probe, it fluoresces in solution and it fluoresces on the membrane. In case of a probe which has closed and open structure, you have fluorescence on the membrane and not in the solution. And why this is important? This is important for in vivo imaging, because when you inject inside the mice, you cannot wash out the probe which did not react. So you want to detect it specifically in the target. And so therefore what we did,
okay, I think we have a connection. Yeah, we have connection problem. Yeah. Okay, you're back now.
It's okay?
Yeah, it's okay now.
Yeah. Okay, yeah, it tells me something about connection, yeah. But you hear me well now? Okay.
Yes, yes.
Okay, so now this targeting ligand should drive us to the receptor, and when we inject into mice, and this is lactating mice, which actually overexpresses in the mammary glands, it overexpresses G protein couple receptor. When we injected this guy into mice, we could see actually mammary glands quite specifically. When we add a competitive ligand, carbitocin, together with the probe, it displaces it and you don't see this image. So it really is specific to the receptor directly in live mice. And in fact, we also took a control mice, which is not a lactating mice, which does not overexpress receptor. And we could see that there is no targeting. So in fact, we could image naturally expressed receptors related to physiological state, thanks to this near-infrared turn-on system. But in fact, this dimeric system was quite universal, and we could even apply it for detection of nucleic acids. It can be opening up in nucleic acids, and this approach is turned out to be very useful in cells as well. But for nucleic acids, what is known? You have many different approaches. You have intercalating dyes like DAPI, which stays nucleic acids, not specifically. You have fluorescence in situ hybridization. You have also some proteins that bind specifically certain nucleic acids. And you have the approach, which we were interested, is based on aptamer and this fluorogenic dye. It was GFP-like molecule originally reported, which binds to a specific binding site of aptamer. And so it's called so-called spinach. But we wanted to do better, much brighter. So we took a rhodamine dye. For rhodamine dye, there was already known aptamer, and was monomeric aptamer and monomeric dye. And we created a dimer like this. And so when we create a dimer, what we need to evolve the structure. So using so-called directed evolution. So this was done with our collaborators, biotechnologists, who mutated this structure and created thousands and actually millions of those different nucleic acids. And then we had to fish out the structures which are fluorescent. And what we found out, that the nature during the mutation created a dimer structure. And this dimer structure turned on the probe. So again, the dimer shows a typical excitonic of the aggregate, of Ash aggregate. And after binding to the aptamer, it becomes a normal molecular state and the fluorescence turns on. So when you put it inside the cell, you can visualize it directly inside the cell. So here it's a RNA, which expresses an aptamer. And then we add the dye goes and binds the aptamer, it lights up. And it colocalizes with GFP because it's co-expressed just as a control. And you don't see anything if you don't express the aptamer. And what is important is that this dye is very stable because it is based on rhodamine. Rhodamine is highly stable structure, fluorophore. And when you compare with previously reported fluorogens, which are actually not based on dimer, but they are actually rotation-based structures, which are not so photostable. It's much more photostable than others. So we gained with amount of photons per cell. So I finished my part on molecular probes, and I gave you some examples of solvatochromic probes, molecular rotors, and fluorogenic systems based on dimers. Where you could have numerous applications in the imaging of membranes, in detection of apoptosis, and also for imaging and detection of proteins and nucleic acids. Now I go to the nanoparticles. So in fact, the interest in nanoparticles is that, if you notice, I was going from dyes, from monomeric dyes to dimeric dyes to increase the brightness. We want to increase the brightness by combining fluorophores together because the brightness brings us sensitivity and resolution. And you have many different nanoparticles. And their most common ones are semiconductor quantum dots in organic systems, which dominated actually the field for last 20 years. But then you have many new structures like dye-doped silicon nanoparticles, metal nanoclusters, carbon dots. You have a very intense research on aggregation-induced emission nanoparticles, conjugated polymers, nanoparticles, etc. But we were interested in dye-loaded polymeric structures. So why? Because you actually can choose the matrix you want, you can choose the dye you want, and like this you can vary the color. The brightness can be very high because you can put hundreds of dyes per particle. And like this, your brightness, which is defined by quantum yield, extinction coefficient, the number of dyes can be very high. But here, when you design polymeric particles with dyes, you need to address three challenges. So the first one, how to make small particles out of polymers. Usually polymers, when you nanoprecipitate them or formulate them, you give big particles. How to make small particles? Then, when you put dyes into the polymer at high concentration, the problem that they aggregate. I showed you example of a dimer, which actually makes an Nash aggregate, it's non-emissive. So how to address this problem? And then how to make probes out of these structures, because these structures are very big, and how to create couple a molecular recognition with some change in the emission, for example, by energy transfer. How to do it? So these three issues we addressed during our like last 6-7 years of research. So first, how to make small particles, and this we actually addressed at the very early studies, where we actually found out that a single charge per polymer can drive a nanoprecipitation process into small particles. In fact, the nanoprecipitation is when you take a good solvent like acetonitrile, dissolve a polymer, and precipitate in water. It's a called Uso effect or Pastis effect, is that it goes to the highly polar environment where polymer is not soluble and it precipitates. But when you have a single charge per polymer, charge prevents the aggregation. It forms small particles, and these small particles are stable. And this we showed by taking, for example, PLGA polymer and adding 1 or 2 charges per polymer chain, or PMMA and 1 or 2 charges, also PCl, and we could see the neutral polymer 100 nanometer or more, and the charged polymer goes below 20 nanometers. And so we could obtain very small particles by a very simple protocol. And in fact, by changing the number of charges per polymer chain, you can change the size. So you can tune the size by the number of charges per polymer chain. And by simple nanoprecipitation, we actually could go down to 7 nanometer, almost the size of proteins. How to prevent self-quenching? So in nature, we know photosynthetic complexes. There, those porphyrin molecules, they are separated, well organized in space. They are separated from aggregation by proteins, and this allows a very efficient energy transfer to a synthetic center, which leads to life actually on the planet. And chemists, what they did different approaches. For example, in case of dendrimers, they could add rigid linkers like this and organize fluorophores in space. The aggregation-induced emission approach, which is blooming now, it uses different moieties which are distorted, like this tetraphenylethylene structure with fluorophore. It gives you aggregation at the solid state. You have a more conventional approach just by adding very bulky groups to a fluorophore. But adding these bulky groups separate in space the fluorophores, and it gives you higher emission in the solid state, preventing aggregation. We were interested in another approach using the conterion. Why? Because most powerful fluorophores are charged. For example, rhodamine, I was already mentioning it. It is highly bright. It's one of the brightest dyes we know, but it aggregates actually because it is flat, but it also because it has this small conterion that helps it to get together. So you have plus, minus, plus, and this sandwich structure makes the aggregate, the H aggregate, which is non-emissive. So we thought we could replace in the rhodamine, a conterion of the rhodamine with a bulky one. And earlier work showed that it's possible for other dyes. So we say, okay, if we use this conterion, maybe we can get the blend with the polymer and get high emission. So to do it, to use it as a spacer, but also maybe because it's highly apolar, we can help encapsulation. So what do? We take a rhodamine perchloride and we exchange conterion with the bulky conterion. And in fact, we tested many conterions. We found out that the magic one is the perfluorinated derivative. We mix it with a polymer, any polymer, in this case it's PLGA, in organic phase. We nanoprecipitate. And what we obtain our particles of different size depending on the polymer. And when we increase the loading, we can see with the conterion, the size is always stable. If you take a perchloride, when you increase the loading, the size increases. And in fact, it's because the encapsulation is not good. It goes to the surface and the particles aggregate. So when you increase the loading and you look at the fluorescence, in case of perchlorate, which means the small conterion, when you increase loading, initially the brightness increases and then it drops, the intensity drops, because it's a self-quenching of dyes. When you use bulky conterion, the fluorescence intensity increases and it continues increasing with loading, and the quantum yield is much higher. So the conterion works as a spacer that prevents the aggregation. And now we compare with quantum dots. What do? We place the particles on the surface of glass, and after we look at them at the single particle level, though these little dots are our particles, and the height of these dots represent the brightness of the particles. And so when we compare our highly loaded particles with quantum dots, we see that they are much brighter than quantum dots. And then actually in 2014, they were 6 times brighter, and now in 2021, we have particles which are 100 times brighter than quantum dots for the same size of quantum dots. And what is this magic effect of conterion? So for that, we studied a variety of different sizes of conterions. Perchloride I already mentioned, but then we say we tried perfluoro. So we took different structures, tetraphenyl borate and others, and then what we show that starting from this structure and larger, we have a jump in the quantum yield. Fluorescence quantum yield goes up rapidly, and it also increases the encapsulation efficiency, starting from these bulky guys. So it has a double effect: quantum yield spacing and the encapsulation. In fact, when the small counterions are used, the dye goes on the surface, and if it goes inside, it's aggregating. And when we use bulky counterion, encapsulation is good, and there is no aggregation. This approach actually was also extended to other dyes. For example, cyanide dyes of different color. You can go from green to near-infrared dye, and using different counterions. In this case, we use the bulkiest one. So now we go to biology. So what happens if you put particles into the cells? What will happen? In fact, the primary thing, we wanted to study something inside the cytosol of the cell. But to get inside the cytosol is tricky. So what we wanted to do is something directly to inject particles inside the cell. And what we found out that the size matters a lot here. So if you take 32 and 17 nanometer particles, you get a totally different result. In case of 32 nanometer particles, when you inject, the particles don't move. You just see the spot of injection. If you take a very small particles, you see of the spot of injection, and then you see the particles spread. So the size actually allows small size particles migrate rapidly inside the cell. Then the size is very important. Now, if you just simply add the particles to the cells, what happens? Particles eat them. And how do they eat them? By endocytosis. So the particles internalize, and you can see these red dots are particles in endosomes. This is just a green stain of the membrane. You can colocalize them with lysosome, with lysotracker. You can see that they are in endosomes and lysosomes. So, and these systems are not toxic. We check with cytotoxicity, and we thought, okay, if we can internalize them so easily into the cell, can we use that for staining of the cells? And we did more than that. As we have particles of different color, we can use blue, green, and red particles. And what we can do, we can actually load them by combining blue, green, and red in different combination, like a printer prints on the paper any color you want by mixing inks. We say we will mix particles, not the dyes, the particles. And by combining, for example, you take the blue, and blue particles give you blue cells, red cells, but then red and blue give you these magenta ones. And then you can do different combinations. Red and green combination will give you green, will be yellow. All three colors give you white. And like this, we generated like 13 different codes. In fact, you can generate more codes by combining different color. And when the cell divides, it keeps the color code. You see here, the color code is preserved, and it divides and it keeps it. So what it allows us to do, you can actually color code different cell lines, then you can mix them, you can co-culture them, and you can recognize them by the color. You can actually color code a 3D cell structure, then let the cells migrate, and you can recognize them on the surface by the color code. You can even inject those cells color-coded in a live animal like zebrafish, and you can track them inside the zebrafish embryo. These are cancer cells. There are 6 different cancer cell lines. You can recognize them inside the cell. And this is in a live tissue. Okay, so this was for imaging application. This is a final part of my talk. How about making sensors? And for sensors, the motivation of making sensors based on nanoparticles is a point-of-care diagnostics. What is the challenge in point-of-care diagnostics? It detects targets like RNA. So now we all face the problem of coronavirus, and we want to detect RNA of coronavirus or RNA of a cancer marker. These guys present at such low concentrations, picomolar. Fluorescent techniques, unfortunately, allows us to detect only nanomolar concentrations. And nanomolar concentration is irrelevant to clinical applications. So we say, okay, how about nanoparticles? They're so bright, maybe they can allow us to reach those nanomolar, picomolar concentrations and below because of their brightness. But for that, for example, if you make a FRET assay, in a molecular FRET assay, you have a donor and acceptor, 2-5 nanometer, you have perfect FRET, then you break it, you have a sensing. In case of particles, it's not obvious because particle is big, it's 10 nanometer size. So how can you FRET from a 10 nanometer particle if the distance is 5 nanometer? If you go above 10 nanometer, you have no FRET. This is the first rule. And it is known for quantum dots, you need a lot of acceptors to transfer energy because of the problem that it's too big. And in fact, by serendipity, we found out that our particles are not usual. What they do, they blink. They actually blink like quantum dots. But blinking is happening for many, many dyes per particle. So here the blinking happens for 100 dyes per particle. And what we check, if you, for 100 dyes you have blinking, for 500 dyes you have even better blinking. What does this mean? That all dyes behave like one ensemble, they go up and down, up and down. Why? In fact, our physical studies, I don't have time to explain, show that actually the dyes communicate. The energy migrates freely and rapidly within the dyes. And then when you have a dark state, a dark dye, it's a sink. All energy goes into it. So in fact, it's a kind of a light harvesting system. Like I explained you before, in nature, in a leaf, energy comes to one of the fluorophore, then the energy migrates, and then it comes and consumed somewhere in the dark state or in the reaction center. So they say, okay, we could maybe build a light harvesting nano antenna based on our donor dye acceptor and acceptor. And this is what we did. We make a particle which contains about 10,000 donor dyes and only single acceptor. And when we record the spectrum with acceptor, we see a very already efficient FRET, like 30% FRET for about 10,000, okay, 6,000 donor per one acceptor. So now what happened is that a huge amount of dyes give energy to a single species. This creates a light harvesting amplification, also called antenna effect. And this antenna effect grows with the size. And in fact, we could reach more than 1,000 amplification, so that acceptor emits 1,000 times more than if it is directly excited. This amplification is higher than even the systems like plasmonics. And this is the best system reported today based on plasmonics, where you place a fluorophore between two gold beads, 300. We got more than 1,000. So this simple organic system can amplify amplify light, amplify the brightness. Now, how can we use it in sensing? We can do it in a FRET assay. So what we did, we functionalized particle with nucleic acids, and we hybridized an acceptor at the surface of particle. Hybridized with the shortest sequence. When we detect a target RNA or DNA, it displaces the acceptor. And like this, we lose the acceptor at the surface. So what happened? Initially, you have FRET. And then we displace acceptor. We have no FRET. So the particle changed from red to green. So does it work? First, we need to synthesize it. Synthesis was not obvious because we need to first place a reactive group at the surface and couple with the DNA. DNA is water soluble and polymer is not water soluble. And vice versa, you can polymer is soluble in organic solvent, but DNA is not soluble in organic solvent. So therefore, what we did, we synthesized particle with azide, and azide was exposed using carboxylide. So this was an original approach we found that worked. And then we did click chemistry, couple-free click chemistry with DBCO DNA. So we place DNA on the surface of particle, then we hybridize acceptor. So when we hybridize acceptor, we observe a nice FRET. So here you have about 20 acceptors per particle, which contain 3,000 donors. We have a nice FRET. Then we add the DNA, which is complementary, which is supposed to displace, and we lose FRET. It works well, and with mutants, they don't give this effect. So it's okay. It's specific to sequence. The more interesting, when you put it on the surface and you record individually acceptor and donor. And what you can see in acceptor channel, you can nicely see the red emission of acceptor. And in the donor, you see the green donor. When you add the target, you see that acceptor disappears and the color switches from orange to green. So at the single particle level, we could see the hybridization processes. When we checked their sensitivity, we reached already sub-picomolar concentration, 0.25 picomolar, which is, we are in the attomole regime of detection. So we start to be already relevant for applications, and the system works in serum. So we now go to point of care diagnostics. And in fact, there is a startup company which is created to develop those systems for diagnostics. But we wanted further challenge and detect actually a single RNA or DNA molecule. So we had to adjust the size of the particle and the dye loading, etc. So we were having 1,000 donors for 20 nanometer particle. And we had a high loading, 50% with respect to polymer. And now what we achieve is that a single hybridization on the surface of a single acceptor, you have 50% of red. So when you deposit this on the surface, you have either green or red-orange particles. In fact, it's a digital, either no acceptor or there is acceptor. And the particles either green or they fret or not fret. And when you do a video of those particles over time, you could actually see the single hybridization events where the red, because the single molecule binds to their cell, to the particle, and those which unbinds, it goes back to green. So this blinking is detection of a single RNA or single DNA, and this is thanks to the highly amplified particles that can amplify the system. And the last slides are about when the system changes the color, we can use actually mobile phone to detect it, because changing the color, we can see in color photography. So we could use a cheap or a color camera, RGB camera of a mobile phone, and then having donor in the green channel and red in the red channel, we could actually distinguish, detect RNA or DNA. And this is what we did. We took a mobile phone, but okay, our system is still very big. We attached the mobile phone to the microscope. And then we do experiments like we did before. We detect the green and the red channel on the RGB camera of the mobile phone. And we could actually see in the real color images, it's not very well seen, but when you split the channels and you make a ratio, you can nicely see the change in the color of the particles on hybridization. So we could use a mobile phone, just we need to miniaturize this microscope to get a system that works in a mobile system. But you have so many gadgets already on the internet to have those attachment to the mobile phone. So now I come to the conclusion. We propose an approach to prepare polymeric particles using counterion that prevents dye self-quenching to get bright system. We develop actually charge control nanoprecipitation of polymers that allows you to go to very small sizes, actually to tune them. By developing particles of different color, we could track cells and code them and track them. So we developed surface chemistry of those polymeric particles, especially click chemistry, which allow us to prepare DNA functionalized nanoparticles. In fact, we observe a unique phenomenon of light harvesting, which amplifies light, which can be used to design nanoprobes, FRET systems, which are highly promising for point-of-care diagnostics, probably, for example, to complement the PCR-based detection of nucleic acids and imaging also in the cells. And so I have to thank my team, as many people worked on. So here in bold, I selected those who contributed most, but there were many projects, so I will not talk individually, but there are senior researchers and there are PhDs and postdocs that work hard. We have collaborations within our university with Biophotonics unit. We have multiple collaborators on different fields as well. I have to thank them, would like to thank them. And I thank the ERC grant, which actually boosted research in our team and other grants at university and CNRS. And I thank you very much for your patience and for your attention.
Thank you so much. Wow, this a lot of materials and a lot of very interesting science, and I'm so very excited that the patent has been filed, and there's a startup company to extend your discovery to the commercialization phase. Congratulations so much.
Thank you.
So now I'd like to invite the audience for questions, please. So while they get ready, maybe I will start. Yeah, sorry. Yes. Hi, Pengkang, yes.
Can you hear me?
Yes, I can hear you.
Hey, Andrey, so nice to see you here. So the wonderful the presentations. So finally can experience your nice fluorescence chemistry. Yeah, brilliant. Congratulations.
Thank
I
you
have
very
a
much.
couple the questions regarding to your very interesting the system. So for your actually these few days I just read your Nature Chemical Biology paper. So the spinach the system.
Yeah.
So you have to like a dumbbell the system. So for your dimer the molecule, if you wanted to insert into this DNA the pocket, so you need control the distance, or I believe your dimer molecule simultaneously fit into the domain, then you can observe the fluorescence, right?
Exactly. So in fact, we did not design ourselves their aptamer. The nature designed it for us because we did a directed evolution. However, what we designed is the dimer die. So we need an optimal distance here in order to ensure their formation of the dimer. If the distance is too long, the dyes will not find each other and will not aggregate. If the distance is too short, they aggregate all the time, and so you will not have fluorescence. Now, when we apply this dimer to their system, to this soup of many mutants, what we select the most optimal one. I did not put the structure, I'm sorry. In fact, I should have put the structure of the aptamer. The aptamer is practically a dimeric structure of this, with the distancing which matches perfectly their two pockets of these two guys. So in fact, it is absolute must that the opening would fit. And if the distance would be too long, it would not work. You are totally right. And in this case, we also gain affinity because you have this avidity approach that you bind two at the same time.
Yes, so that for me make more sense. So you can precisely optimize the distance to fit into the pocket to observe the fluorescence. But is that possible? So when you design the distance, somehow your dimer molecule can only go to one pocket, the other diffuse away. Is that possible?
Yes, I think you have a possibility like this. Probably it will have a kind of low affinity. If you increase concentration, I think it will happen. You will have two dyes binding at the same time. But I think this single dye binding will happen for some of those mutants that we tried, but we did not select them because of low affinity and because they will have a low intensity. When you have a dimer which binds only with one end, I think the affinity will be really low and the turn-on will be low. But what we did actually, because chemists can control, we start varying the structure of these guys, changing the groups here. And we find out that you can improve chemically the structure and to fit better into the pocket. So you don't need to ask nature to redesign, you can redesign yourself and make better structures.
Well, that is so nice, very smart the design. But I'm sure, so next step, so you are trying to make this for live cell, let's see, RNA detection or the imaging in tumor, right?
Yes, in fact, we already showed in our first example of this in this Nature Chem Bio paper, it works, but it's not working perfectly yet. Why? Because this molecule is big, but also because this molecule has sulfonate groups. And these are very polar groups, and their internalization into the cell is slow. And so what we do, we now modify the structure. For example, we modified with carboxylate, and now this article is under revision. So we found out that replacement with carboxylate drastically improves their permeability into the cell. And so we can improve their cellular imaging by redesigning the dye.
Nice. Yeah.
And so you need to express an RNA of interest with this tag, and then you can follow the RNA. It's especially interesting in viral research. There are many people working on viruses. They contact us because they want to track virus with the RNA tag.
Yeah.
Yeah, very nice. So that is all my the question. Thank you so much for briefing. And hopefully, so COVID-19 quickly disappear, and sometimes you can visit us physically in Singapore.
Yes, thank you. Yeah, it would be great. I never been to Singapore.
Yeah, welcome. Last time I visit Loic, the Champagne, so just happened. So you have you're on leave for your holiday. So but hopefully in the future.
Yes, me too.
Yeah.
Thank you.
All right, thank you for briefing.
Thank you.
Thank you.
Any other questions? So Andre, for your DNA detection, for the 1,000 times amplification, I'm just wondering whether for your the probe that is on the surface, is it dependent on the length of the base pair? So what is the typical base pair, the number of base pair that you employ on the surface?
Yeah. So here we functionalize a particle of 40 nanometer with about 100 nucleic acids per particle, 100 to 200. After that, what we do, we hybridize an acceptor just next to the particle. So here there is only when you in the design you have just one spacer of one base. And then you have an acceptor. So the acceptor is just really near the surface. If you place it more far, the efficiency will drop because it's a strong distance dependence. Yeah. So therefore, we actually need to design, if you see the acceptor is here, it's not here, it's really close. And that was absolute must. And you also need to control the number of those acceptors per particle, because if you don't have enough, you will not have a good thread. So you need to design the system with a controlled number of acceptors per particle.
Yeah. So what about your capture sequence? Your capture sequence, is it also base pair specific, as in?
Yes. Yeah, exactly. So the capture sequence should be complementary to the target.
Yeah, sure.
And so, and usually it should be around 20 mer. So 20 mer, it gives you a perfect specificity to the target. And for example, in this case, the FRET acceptor is conjugated or hybridized with a shorter sequence.
Okay.
So it's about 12.
12.
And so, so what happens that the longer sequence of the target will displace this acceptor because their hybridization with the longer sequence is efficient. And so it will displaces the shorter sequence, you see?
Yeah, so the total length is about 32 base pair, 32 mer.
No, actually the total length here is 20, and this short one is here hybridized of 12. So this is 12 plus 8, 8 or actually it can be 22 up to 22. So it's 12 plus 10.
Okay.
So you see, and so then displacement gives you the changing frame. Yeah.
Okay, thank you. Hi, Edwin, you have a question?
Hello, Andre.
Hello, Edwin.
Hello, I'm trying to turn on my video, but it's not working. How are you, Andre?
I'm sure you're hiding yourself.
That's right. Every time with you around.
But I recognize your voice. It's great to hear you.
You can recognize my voice?
Of course, I recognize your voice. Yes.
So everything is good?
Yes, everything is good. You know, Edwin, I used your paper on anisotropy to prove that we have there. Oh, yes. Oh, but you did not change. It's the same as during the postdoc time. Yeah. So, Edwin, actually, I have to say that we really were happy to use your article to prove this migration of energy because you had the work on the anisotropy studies,
right?
And we use this anisotropy to prove actually that the energy migrates, and so their anisotropy drops. And so, yeah. So it was the world is smaller.
Yeah. So it was useful.
Of course.
So, Andre, I have a question. You know the one where you did your energy harvesting, right? And then after that, everything migrates and then goes to your this one. So did you find that if you have more of the chromophore, your fluorescence actually this more energy gets sent over to your energy acceptor?
So in fact, what happens is that I do not show it here, but when you create this system and when you have a few donors, for example, now you have like 10,000. If you make, for example, 100, you actually have no thread at all. When you put 1,000, you have a little thread. And when you put 10,000, you have a huge thread, which is not at all logical at the beginning, because you put so many donors and it actually gives you more transfer.
I would think that there will be self-quenching if you have too many.
Yes, so in fact, the trick is that when you put many of them, they self-quench. That's why usually people don't observe this phenomenon. However, in this case, you have the donor dye with this magic counterion. So when you have these two guys together, there is no self-quenching. And so you can actually load many donors, which emit still with quantum yield of about 50%. And so you have such high density of donor dyes in the particle, and they are close to each other, and they emit with a quantum yield of 50%. That actually lets the system relax very slowly. So you have an energy which migrates within 4 nanoseconds, and the speed of migration is actually down to 30 femtoseconds. So the hopping is on the femtosecond regime.
It's very fast.
So it's super fast. So which means that actually energy migrates within the particle within 4 nanoseconds. All fluorophores were solicited. So they were all getting excited. And then if you have acceptor, it goes there. However, if you decrease number of dyes, you increase distance between dyes. And they stop communicating to each other. And so there is no more transfer.
Do you think it's still faster, or do you think it's what they always say?
I think it's not faster. It's actually kind of
some sort of a coherent, like those things that they talk about in photosynthesis. Is that those?
Exactly.
Quantum.
Yes, this is. Yeah. And it's exactly that. I think we are no more in the first regime because their energy transfer is too fast. And we have actually, I talk about 30 femtosecond. We have a component 30 femtosecond. You have longer components, but this 30 femtosecond is remarkable. And I think it is their coherent part because it's probably instantaneous. And as people are talking about coherent transfer in light harvesting systems, and there the distance between porphyrins is even longer than when we have here. And so therefore, I'm quite sure we have the same. And in fact, now we are running a project to understand whether it's coherent or not. But any ideas how to prove coherent transfer in these systems are welcome, actually.
Can be challenging.
But you're right. It's really a kind of coherent system because it has so unusual efficiency.
Yeah, because I noticed. Yeah, so it's really acting really like a light harvesting system.
Yeah. We made a synthetic version of this.
Right. Very nice. Yeah.
Thank you, Edwin.
Hey, thank you so much, Andrey. Hey, so we should, once this COVID is over, we should meet up. Either in Strasbourg, or the next time when I invite you here, you must make sure you stop buying houses and start to come here.
I already bought. It's true that I had a really bad excuse, but I was signing practically the purchase of apartment during your meeting. I'm really sorry not to come. It was
no problem.
I saw the photos from the meeting. I was very, very upset. I was happy for you, but not for myself.
Right. Next time, there's always next time. Okay. Maybe I'll leave I'll get other people to ask questions.
Thank you, Edwin. Thank you.
Thank you, Edwin.
We can take 1 or 2 more questions. All right, we can take 1 or 2 more questions. Any more questions from the audience, please?
Hi, Dr. Andrey. I'm just curious regarding this light harvesting nano antenna. How do you accurately measure how many donor groups have been encapsulated within each polymer, and how do you determine, let's say, if the if the release of the FRET acceptor is a competitive or non-competitive sort of direction. So as time progresses, does the FRET get restored?
Yeah. But when you mention the last point for the acceptor, repeating once again.
Oh, right. So looking at the time-lapse video you had just now, it sort of seems like there's some sort of competitive binding between the target molecule and the FRET.
Yeah.
So over time, the FRET gets restored and it goes on and off.
Yeah. It's right. Yeah. Now I understand. Okay. So I can comment on that. I don't have the data here on single molecule except one here, but let's start step by step. So how do we know the number of dyes per particle, donor dyes? So here it's actually quite tricky. What we do, we prepare nanoparticles with a known concentration of dye per polymer. Then what we do electron microscopy to measure the size of particles. So based on the size of particles, we can calculate the mass of the particle. So from mass of the particle, we can calculate the number of moles of dye, because the encapsulation efficiency is 100%. It's 99 actually percent. We measure that using dialysis. We know that encapsulation efficiency is quantitative. So as you know the mass of the particle, you can access the number of dyes by simply calculating the mass of particle. This is so for the donor dyes. For the acceptor dyes, it's different. So for the acceptor dyes, first for the quantity of acceptor dyes, when you encapsulate or put them on the surface, you can always measure absorption and you can know the ratio, this or this. It's a tricky because the difference is very large between number here and here, but you can still calculate when the ratio is 1 to 100 or 1 to 500, you can do this. So like this, we can make a ratio of donor to acceptor. We can calculate number of acceptors. However, this is not working in case of this light harvesting system when you have a ratio of 1 to 10,000. So what we did a single molecule measurements where we did a trace, fluorescence trace of the acceptor. And what we observed is very similar to what you can see here, but I don't have single molecule trace. But actually what happened, it happened like this. You see the particle, which is red initially, then it becomes green. And what happened is that when the acceptor drops, the donor goes up. And you have these processes. So if you have a single step process, this means that you have a single acceptor. But if you have multi-step acceptor bleaching, you have multiple acceptors. And in fact, that was one of the reviewers asked us this question. Please quantify number of acceptors in your system. And we did this single step bleaching and we counted, and statistically in this light harvesting complex, we had a single acceptor per particle, and we had also 2 acceptors per particle, but this was much more rare, and we had no dye. It's statistical. In this process, yes, it is a kind of competitive, but in fact here, in this experiment, we are hybridizing a very short DNA to their long DNA. And very short means like it's an 8-mer. And 8-mer binds and unbinds very fast. And so therefore the acceptor comes, FRET, and then it goes back and there is no FRET. And so this is a dynamic process. That's why the particle can do 2 times or 3 times red. You see red, green, and then it becomes red again. Again, you see it becomes red again because it's many times. It's known, for example, DNA paint. I don't know if you hear about this. This is a reversible binding. It's used for super resolution. Here we use it for reversible FRET to see the single process. But otherwise, yeah, the phenomena are happening at the single acceptor level.
All right. That makes sense. All right, thank you so much.
Yeah, you're welcome.
We can take one more last question. Anyone? Okay, so if not, let us thank Dr. Andrey again for this wonderful talk. Yeah, So hope to keep in touch with you, and I'm sure there are many friends and faculty here who would like to invite you physically over.
Thank you very much. Yeah, thank you very much, and thank you for this discussion.
Yeah,
and for your interest. And so it's a pleasure to meet you guys, and also to meet my old friend Edwin. Thank you for joining. And so we keep in touch.
Yes, let's keep in touch, and please take care.
Yeah, take care.
Thank you.
See you.
Thank you, everyone. Bye-bye.
Bye-bye.
Bye.
Bye-bye.